everybody. We'll just begin with the session. I'll share my screen. Yeah. Um, hello everybody. Welcome back to this particular session for this course, Basics of Neurobiology. Herein we will be dealing with the sensory modalities of the brain, right? So we'll delve into deeper details regarding the sensory modalities. What are the sense organs? What is the role of the brain in perceiving whatever information we are getting from these sense organs, right? For those of you who are new to this lecture, you are directly landing on this lecture. Let me just briefly introduce myself. So I am Arveen. I am a PhD scholar from Department of Biological Sciences and Bioengineering at IIT Kanpur. Together with my funding agency, my institute and ISSS, we are bringing you this particular series. And this is lecture number 8 for this particular course, that is Basics of Neurobiology, right? So earlier in this course, we have already understood what is the historical perspective, how we came to know about our current understanding of the brain, right? How people have done experiments and reached to the status of knowledge that we have about the brain as of now, right? We have gone through that, the primitive, neuroscience, neurobiology history, as well as the contemporary history. Also, along with that, we have discussed what is the brain, what is the nervous system, what are the parts of the nervous system, what are the various parts of the brain, what is the cellular unit of the brain, what is the kinds of cells that are present in the brain, etc. Right? What is the cross morphology of the neuron as well as the entire brain. So all this also we have studied. We have also studied the functional, how there are different functionally organized regions that are present in the brain, how they cross talk between one another. A little bit about that also we have discussed. Then we have also, looked at the techniques that have been used to study the brain, and they are also clinically and medically relevant because that is actively used in by the physicians, by the doctors for the clinical testing of the brain also. Thereafter, we have studied the development of brain. How does that happen from the level of embryogenesis right to an adult brain? Then, finally, we want to end our discussion with the sense organs and how various sense organs are basically linked to various modalities in the brain, right? How does the brain respond? How does it perceive different senses, etc. Right? So, as we begin with this particular lecture, let me remind you of the basic senses that we have, right? What are the major sense organs of our body? So, if you recall, we have studied this back when we were in school, right? That the basic sense organs that are there in our body are eyes for visual perception, ear for auditory perception, nose for olfactory perception, tongue for gustatory perception, and skin or touch. Not skin, touch for, physical perception or spatial, perception, something like that. So these are the 5 major senses that we are aware of. Apart from this, earlier in this unit, we have studied that there is another kind of sense of knowledge in our brain, that where our hand, where our limbs are, where in our body is, our leg, where in our body is my left hand's third finger, and all these things, right? So that our body is aware about their spatial being of its other body parts, right? That kind of uh, sense, that is also a kind of sensory perception, that is known as proprioception, because it is kind of the stimulus is from within the body. It is not from outside the body, it is internal, right? Also, if a body is heating up, for example, we have fever, so there is a temperature that we are getting continuously. So that kind of a perception is also going to be a part of proprioception, right? So these are the major sensory modalities that we have, or sensory sense organs that we have, right? Now, how is the brain involved in actual sensing, right? That is what we are going to deal with this in this particular unit, right? So let us just begin. As you know, some of these I have just spoken about. Vision is the, response that is generated through the eyes and the brain, and it results in seeing or viewing an object, right? Similarly, smell is a response that is generated when some kind of a chemical molecule, volatile in nature, it reaches our odor receptors, and they basically sense that this molecule is there. They take this information higher into the brain. The brain responds to whether it knows what it is, whether it doesn't know what it is, or what it is, that particular odor. It responds accordingly, and then you judge based on that whether what is the smell, whether you are familiar with that smell, whether you like that smell, whether you don't like that smell, etc. Right? Then taste is another sense, right? Over here, the major organ responsible for taste is the tongue, right? So this muscle has a lot of taste receptors or gustatory receptors, which look like this, that are present. And there are different kinds of taste that again they can distinguish in, right? So that is how we know whether this particular nacho, it tastes more cheesy or it tastes more, spicy, or it has some kinds of different herbs like oreganos and all, right? That is how we get to know, because there are different set of combinations of these receptors that are stimulated on eating various kinds of food, right? Or the same kind of food that has different taste also, right? So you can distinguish between maybe lentil that was, or a dal that was made in your home versus a dal that was made in your mess, right? So that kind of distinction we know, right? The fourth kind is touch, right? So that is what we were talking. It is one of the perceptions which is there in the entire body, right? So there are touch sensitive receptors in our entire body, but most of them are localized in our hands, right? Because the maximum tactile sensation that we experience on a daily basis, that is via this touch receptors that are present on the hand, right? So again, we will study that in the hand alone, there are different kinds of these touch receptors that are present, and they have a little different function also with respect to one another. Okay. Then there are thermal senses, right? Now, thermal senses could be the sense of feeling hot or feeling cold, right? So that is also some kind of a sense. Mostly the stimulus can be external, right? So somebody has turned on the AC and you're sitting right in front of it. So you start feeling cold because you have not worn clothes according to, that temperature. Right, so you start feeling cold. So that is also a sense, because you're some kind of mechanical or some kind of change is perceived by your body. Similarly, if you go out in scorching heat, or you touch an induction pan, right, or you go near a very hot candle, a hot burning candle, you will feel some kind of sensation, right? That will maybe lead you to retract your hand, or it will tell you that, oh, it is cold outside, so yes, over here I'm getting some warmth, so I'll stay over here, something like that, right? Then pain is another kind of sense that is there, right? So pain is also a sense, although it has also, okay, that is a separate topic, we'll not speak about it right now. So pain is a sensation of some kind of harm that is being caused to your body, right? Then there's hearing. So hearing happens via ears, right? Your ears are responsible for hearing a sound, right? So what is the kind of stimulus over here? It is basically a frequency or a wave change in the atmospheric air, right? So if I'm speaking, how can you hear? Because I'm speaking something that is being picked up by that whatever is being emitted out of my mouth, it is causing some kind of frequency change in the air that is surrounding me, right? Because of that frequency change, the microphone that is there on my system can detect that frequency change, and it is able to capture whatever I am saying, right? And you, on the other hand, because you are sitting in front of another system or a mobile or a laptop, right? So the speaker over there is ejecting the same frequency from at which I am speaking, right? So it is ejecting the same frequency, which is basically causing some kind of changes in the air around you, so that those changes are then being picked up by your ear, and then your ear can convert this stimulus of this air pressure difference into a chemical stimulus, which can further be transmitted to the brain, right? So that is how hearing occurs. Then balance is also a feature of the ear, right? Ear and the eustachian tube that is present. So the internal ear has some organ that is known as semicircular canals, which are responsible for the sense of balance, right? So if you stand up, you can stand straight, right? You are not falling down, although the gravity might be pulling you down, but you can stand straight, right? You are not drooping on one side or the other side, or towards forward or backward, right? So that is not happening because there is some kind of balance that is maintained in our body, right? In cases of vertigo, sometimes this balance is lost or it is disturbed momentarily due to some external factors and conditions, due to which a person tends to fall or tends to fade, right? And then there's proprioception, as I told you. It is basically how our body judges where our other organs of the body are, right? So it is just a sense of knowing that I know I am not consciously maybe looking at my feet, but I know that I have crossed my legs, right? That is a sense of knowing about my body organs or where in space they are, that I have through internal stimuli, right? My brain knows, although my eyes are not seeing there, right? It might be due to the perception of touch that I'm feeling on my knees because my legs are crossed, right? Something like that. But that is what basically proprioception is, right? The stimuli is generally internal. Then these receptors can broadly be classified into three classes, right? One is a chemoreceptor, the second is a photoreceptor, and the third is a mechanoreceptor, right? So, as the name suggests, chemoreceptor will have, because it is all these are receptors, right? So everything will have a stimulant or a ligand, right? So the stimulant for a chemoreceptor will be a chemical, right? As the name suggests, it will be a chemical. So some chemical molecule will go and bind to this receptor and stimulate it or open it so that it can pass through and it can perform its function, change the signaling, alter some signaling in the cells, and lead to, plethora of changes in our body. The other is the photoreceptor. Now, as the name again suggests, it will be activated or stimulated by a photon, right? So these are logically found in the eye, right? No other body organ of yours is, sensitive to light, as sensitive to light perception as is your eye, right? So if for instance you had lost your visual senses were impaired, or then also up to some extent you will be able to perceive light. You may not be able to see what is there, but you might be able to perceive light, right? But if your eyes were shut, they were completely taped shut, right? And you were basically put in a room, will you be knowing whether it is light or dark, right? You will not, because that is the only organ in your body that can perceive light, right? If there is not even a minuscule particle of photon that is entering your eye, you will not be able to perceive that whether you are in darkness or whether you are in light, right? So that is why these photoreceptors, which are activated by a photon of light, they are generally present in the eyes, right? They are responsible for visual uh, perception. Then comes mechanoreceptor, right? Again, as the name suggests, mechanoreceptor will have a stimulus or it will be activated by a mechanical force or a mechanical response, right? So what will happen over here? Now, for instance, I can expect a mechanoreceptor to be present on the surface of my skin also, right? So that if somebody touches or pokes me on my skin, I know that somebody is poking. Because, and how do I know? Is there some chemical that is being passed on from that person's finger to my skin? No, right? Is there some kind of light activation that is happening? No. Is there some kind of chemical that is being transferred? No, right? What is being transferred? Some mechanical activity, right? So some kind of receptor over here might be sensitive to this mechanical activity, due to which, it is responding, and I know my brain now knows that somebody is poking me, right? Or somebody has hit me, right? So that is a response via that is initiated via mechanoreceptors. Pressure sensing is done by that, right? Even if you are outside, uh, there are a few ways you can know that you are in the midst of a thunderstorm, right? One is that you will see that all the things around you, or all the trees around you rather, are shaking violently. The leaves are shaking violently. You will hear that the because the leaves are moving violently, and because the wind flow is so much that you will hear a lot of things getting displaced from their location, falling here and there, right? So you will hear that also. But at the same time, if you are not able to do both of these things, you will also, if you stand in the like in such a way that you are in between the wind that is crossing, right? So the wind that is crossing will also exert some kind of a pressure on your this thing body, and then you will be able to still judge that okay, the wind today is very harsh, right? It is flowing at a higher speed because I can feel it hitting my body that way, right? So that is also a pressure sensation that you have. Then the receptive field of a sensory neuron is basically a spatial domain in the sense organ where stimulation excites or inhibits the neuron, right? So we will read more about receptive fields of various kinds of, sensory modalities as and when we come to it. Over here, just to introduce this concept, what we mean is that if, for instance, I'm using this mouse, right? If I'm using this mouse with my index finger, then index finger will have that receptive field, right? Because I'm touching the mouse only with my index finger, right? But I'm also using the third finger over here to support the mouse, my ring finger, right? So the receptive field will be both in the index finger and the ring finger, right? So it is basically wherever you are getting the sensation, that is basically the area of reception of that stimulus or the receptive field. Then there could be some classification that we can do for these kinds of sensory receptors, right? What are these classifications? Based on which sensory system we are talking about, this classification can either be visual or auditory or vestibular, or somatosensory or gustatory or olfactory, right? Now, what is the modality? What is the stimulus receptor class and the receptor cells that are linked to each one of this? We can look at them briefly, right? So when we are talking about visual sensory system, right, that has to do with the eyes, right? So what is the modality linked to it? Vision, right? What is the function that is happening from this visual sensory system? That we can see. Vision is imparted, right? Thus, the stimulus over here, we know that the receptor class is photoreceptor, right? So the stimulus will be a photon or light, right? And the receptor cells over here are the rods and cones, right? We will read more about them, as you might recall from your school days, that cones are the ones that impart us colored vision. Rods are the one that impart us vision at night, right? Then there is auditory sensory system that is present. It has the modality it is dealing with is hearing. The stimulus that we receive over here is sound, right? And as I said, sound is in form of pressure waves or changes in the air pressure, right? So that is why the receptor class that is over here is the mechanoreceptor, right? And what are the receptor cells that are present? They are the hair cells inside the cochlea of the inner ear, right? Then again, there is another sensory system which is vestibular. Now, this is one that we haven't discussed till now, right? What is the modality linked to this vestibular sensory or system? It is the head motion, right? So whether I'm moving my head like this or up or down, that is basically my vestibular sensory system that is at play. What is the stimulus of the system? Sorry, it is either gravity, right? So, like if my balance is not working, then I might fall down, right? That is due to gravity. Then it is acceleration, right? If you are sitting in a car, all of a sudden, if you accelerate, all of a sudden you will have a jerk-like movement, right? If you are not prepared for it. And normal head motion, right? And what is the receptor class for this one also? It is mechanoreceptors again. Also, the receptor cells are the vestibular hair cells in the vestibular labyrinths as well as cranial and sorry, that is for somatosensory. So, hair cells in vestibular labyrinth is this uh, uh, receptor cells for this vestibular system, right? It is also a part of the ear actually. Then comes the somatosensory system, right? So, somatosensory meaning any other, touch related, perception, proprioception related, pain related sense, right? So if we are talking about this, it is linked with multiple modalities as we can see. And if the first modality that we are going to talk about is touch, right? So the somatosensory system that is linked to this modality that is touch. What is going to be the stimulus? That the skin deformation and motion is going to happen, right? So either I am moving, towards something and I touch it, or something hits my finger so that my finger, like normally it is like this, it is very on a very minor scale, it is deformed, so that I know that something is hitting on it or something is touching it, so that that particular region is not as it is, it is deformed a little, right? So the receptor class over here is also mechanoreceptor, and the receptor cells are the skin cells, right? Then comes proprioception. What is the stimulus for proprioception? Muscle length, muscle force, joint angle, right? What is the receptor class again? Mechanoreceptors, and the receptor cells are muscle spindles and joint capsules, right? Then comes pain, right? What is the stimulus for pain? It is noxious stimuli, which could include thermal, mechanical, as well as chemical stimuli, right? So that is why the receptor classes are also abundant. It could either be chemoreceptor or mechanoreceptor or thermoreceptor. And all the tissues except central nervous system basically have receptor cells that are responsible for pain, right? That is why we say that the brain surgery by itself is not painful, right? Creating that cranial window, opening the skull, that is the painful part. But once you are, even if you are poking needles in your brain, right? Uh, you cannot feel pain over there, right? Whatever response might be generated due to some other neuron getting hit and it firing, that is different. But by itself, you can't feel pain, right? That we cannot say for any other organ. We can't say that if you open up my body and you poke needles in my stomach or you poke needles in my liver, I will not feel pain, right? I will definitely feel pain. But that is not the case of the central nervous system, specifically the brain, right? Then another interesting modality of the somatosensory system is the itch, right? The stimulus for itch is histamine, right? So, for instance, if a mosquito bites you, why do you feel like itching? Because the mosquito basically releases this chemical which is histamine, right? It will prevent the whatever pore it has created from closing all of a sudden, right? And this histamine will cause this itching sensation. So, what is the receptor class for itch? It is chemoreceptor, and the receptor cells are usually the skin, right? Then the final modality under somatosensory system is the visceral, and the stimuli related to that is thermal, mechanical, or chemical stimuli. Again, because they are like multiple classes of stimuli are there. That is why multiple classes of receptors are also there. And the receptor cells include gastrointestinal tract, urinary bladder, as well as lungs, right? Then comes the gustatory sensory system, right? What is the modality? Taste, right? What is the receptor cell? Taste bud on the tongue, right? What is the stimulus? It is chemical stimulus, and that's the receptor class is chemoreceptor. Similarly, olfactory sensory system has is linked to modality smell, which has stimulants in the form of odorants, and the receptor class for these stimulants are also chemoreceptors. Thus, the receptor cells that are present over here are the olfactory sensory neurons that are present, right? So let us begin with the discussion, in-depth discussion of our first sensory modality, that is vision, right? So, although the eyes play an undeniably crucial role in the vision, the brain is involved in actively constructing a visual representation of the world that is not a literal production of the pattern of light falling on the eyes, right? So, that is true. Why are we saying that is true? Because okay, we will understand it with examples that are simpler to grasp, right? So that I have already put in the presentation. Before we get into those examples, let us first, briefly explain to you that there are two classes of the photoreceptors that are present in humans. We will read further upon about them. These two classes are either rods or cones, right? So rods are the ones that are more active during the night, when the cones are actually not that active. That is when the rods are active, while the cones are the color imparting photoreceptors. They are active, more active during the day, right? As you can see, rods, they basically survive over one wavelength itself, but like the range is more, right? The range of wavelengths that they can detect is almost all the wavelengths, right? But they peak at around red green wavelength, right? But in case of cones, there are different kinds of cones that are present: the blue cones, red cones, and green cones. And they have a shorter range as when individually we are comparing them with rods. And in this shorter range also, you can see that they are peaking at different colors. So this is called blue cone because it is peaking at around blue color. It is called green cone because it is peaking at around green color. Red cone is peaking at around yellow color, but it is still present till the far end of the spectrum. That is why it has been labeled as red cone, right? So that is a little bit of a misnomer. Now, what happens is that it is possible that not just one cone is activated at a time, right? If I am seeing a red colored bottle, right, a bottle cap, it is not compulsory that only, red color cone is being activated, right? Why am I seeing this as a different red color and this one as a different red color? Is because there are a number of simultaneous cones that are being activated, and the average of those cones is making me, look at this red and this red differently, right? Similarly, there is another kind of red of my laser pointer that is different from both these kinds of reds, right? So that is what is represented over here, right? So these cones, if only those cones were activated, then we look at these kinds of colors, right? But there is often activation of as many green cones as many red are activated, right? In that case, we will see yellow color maybe, right? Or if all the three cones are equally activated, we will probably see somewhere over here, it will be more like a whitish, creamish color, right? If we see that more of green, more of blue and less of red cones are activated, probably we'll see this kind of a color over here, right? So the color perception will vary based on what is the percentage of what kind of cones that are activated, right? So that is why our eye is actually capable of detecting like millions and millions of colors, because these combinations in which these cones and rods, because there are multiple different kinds of blue cones, multiple kinds of green cones, multiple red cones that are present, and how many of these will be active at what proportion with relation to one another, it will vary, right, with what you are seeing. That is why you will be able to differentiate between multiple kinds of greens, multiple kinds of yellows, multiple kinds of blues, etc. Right? So that is the principle behind it. Also, another important thing to note over here is that the spatial properties of scenes and objects, right? So this is a scene that you can see, and you can see that this one looks more pixelated, this looks less pixelated, and this looks like a clear picture, right? But these two are definitely pixelated, right? And what is the difference we are seeing? This has 20 by 20 pixels, this has 60 by 60 pixels, and this has 400 by 400 pixels, right? So this has a maximum pixels, due to which it looks more like a clear picture. The ones that has the least pixels looks more like, a pixelated or an unclear picture. So what is the outcome we get out of it is that the more the number of neurons or pixels that are present in a particular area, the better image or better resolution we can see with, right? So that means that since we see images mostly like this, right? C1. So we have more neurons that are concentrated in an area, in a particular area, that is all looking at the same thing, all reporting back and forming this particular image. So that is why this high concentration of neurons are responsible for this particular perfect kind of image, right? Maybe in smaller animals that have compound eyes or other, mammals, if the number of neurons was lesser, right? Maybe they will not be really able to see this, but they'll see something like this, right? So that is another like thing that you can keep in mind. Then we come to this discussion about sensation versus perception, right? And then we will talk about visual perception and how it is there, right? So what is sensation? It refers to the effects of a stimulus on a sensory organ, right? Whereas what is perception? It involves elaboration and interpretation of that sensory stimulus based on, for example, knowledge of how objects are structured, right? So sensation is whatever input you are receiving, as it is, that input is sensation. But what is the information you are deriving from that stimulus is basically perception, right? So that is the difference between sensation and perception. For instance, if you look at this image, tell me what you see, right? Have a look at it, tell me what you see. Okay. So if you look over here in this box, you will probably, what you will be seeing is one white colored triangle over one triangle that had a yellow colored border, right? And those are placed on top of, like, the edges of this white colored triangle are overlapping on three different circles, right? So that is probably what most of you will see, right? Because that is how our brain generally perceives this particular image, right? But is it really there? Is that triangle really there, right? What are the individual components that are actually present in this image? The triangle is not really there, right? There are certain components. One is this, the other is this, the third one is this, right? And three, circles that are like this are present, right? Just because they are arranged in this particular format, it looks like it is a triangle overlain on another triangle, and the first triangle also overlain on three circles, right? That is what it looks like. But is it so? Or is it just these pieces that are arranged in a way so that our brain is perceiving it as a triangle, right? So this kind of an illusion, or this particular illusion actually, is known as Kanisa illusion, right? So why does it exist is what we will study in a while, right? Okay, I did not get the laser pointer. Yeah. Let us look at some other kind of these illusions first, right? Similar is the case over here. What do you see? So most people who will spend time and look at this image will see one of these two things, right? Either they will see a shape which is kind of like this hexagonal shape, right? And it has some, crisscross in between, whatever, right? I hope you understood. Or what others might see is a cuboid that is going over here, right? So a cuboid is what most people also might see, right? So, but is there a cuboid? The question remains the same, right? It is actually multiple different kinds of, circles with different kinds of, this white portion in between them that is arranged in a way that it looks like upon these black dots, there was a white circle that was overlain and that is imposing upon those black dots, right? That is what it looks like. So that is the perception of our brain, right? So even if it is not there, that is how our brain is trained to think, right? Because it will try to find sense, it will try to find a pattern in these things, right? So that is a higher level thinking or cognitive abilities that are present in our brain. This one is one of my favorite examples. It is taken from the lecture from one of my professors, Professor Nitin Gupta's lectures, right? And this is how we were taught perception when we were studying neurobiology, right? So if you look at this picture, do you see a pattern or can you make out what this is? I will just pause, try to look at this picture, try to make out what this particular thing is, right? And let's see if you can figure it out. So this is a very good example of a holistic perception or a holistic viewing that is there by the brain, right? So, my bet is that not most of you might have, most of you or all of you might have failed to figure out what this pattern is, right? Now, let us help you a little bit. And all these empty areas that are there between these yellow scattered pieces, say if there was some ink that was spilled in between these areas, right? Then what it will look like is this. Now, can you make out what this yellow colored object is or yellow colored pattern is, right? So if you will see, the yellow areas are exactly the same as this image, right? But just because of this holistic view that our brain has been developed to look at, just because it was overlain, the other areas were overlain with a different kind of, color, and it is patterned in such a way that it looks like there was some ink that has spilled over there or something that has some shape that has been put over there so that it is hiding whatever yellow areas are there, right? That is how our brain is perceiving that this is actually the letter B, the capital B letter in English, right? So I hope you can appreciate the holistic viewing power that our brain has by this particular example, right? If not by the previous two ones. Then let us, to further understand what is the neural circuitry that is being dealt with over here, let us first understand what is the eye, where is the neural layer actually present in the eye, and how is it taking information to the brain, right? So this is what an eye looks like, right? It has three major layers. So apart from that, first let us deal with this part. So it has an iris, that is the colored part of the eye, pupil, that is the black part of the eye, right? Then there is a cornea, that is on top of the eye. There is conjunctiva, that is the kind of transparent lens, transparent region that is covering our eye. There is lens inside this particular region. Then there are certain ciliary body muscles that are helping our eyes remain open, or they are also helping in movement of our eye, right? Blinking, etc. Then there are these three major layers. They are the cornea, the outer layer, the sclera, this middle layer, and the retina, the innermost layer, right? So this innermost layer, that is the retina, has actually the photoreceptors, or the nerve cells that will detect the signal that is coming in the form of photons or light, right? And this entire retinal layer is actually made up of nerve cells only, right? There are different kinds of glial cells and neural cells that are coming and making up this retina. So it is actually a part of the central nervous system. This particular layer, retina, is a part of the central nervous system because it has almost 10 different classes of cells, and most of them are nerve cells, right? Most of them are neurons, sorry, right? So this is the retina, and this retina further contains the photoreceptors also, right? Along with other neurons. So the retina is the internal surface of the eye that contains specialized photoreceptors that convert or transduce light into neural signals, right? So that is what this retina is doing. The photoreceptors are made up of rod cells, which are specialized for low levels of light intensity, such as those found at night, and cone cells, which are more active during the daytime and are specialized for detecting different wavelengths of light, right? So that we have studied earlier when we were looking at that map of the wavelengths that are covered by these RGB cones as well as the rods, right? This is what the retinal layer in transverse section looks like, right? So if we zoom in this particular yellow colored layer, which is the retina, we will see that there are multiple cells that are arranged in this columnar fashion that are actually constituting this retinal layer, right? So you can see over here is the sclera, the sclera and the choroid, right? After this sclera and choroid, from here onward starts the retina. This entire portion is the retina, right? So first come the retinal pigment epithelium, RPE, then come the rods, then cones, then horizontal cell, then bipolar cell, then amacrine cell, then ganglion cell, and then the optic nerve, right? So if you will see, another important feature, right? So this is in this order only. So over here on top most is the optic nerve that is connecting to these ganglion cells. On the topmost layer is a ganglion cell. On the lowermost layer is the retinal pigment epithelium and the rod and the cone, right? So another kind of fascinating feature of this particular kind of perception, or this particular kind of sensation in eye, is that over here, although we all know the light will come in this direction, right? From here, somehow it will come and it will reach different parts of the epithelium, right? Different parts of the retina, right? And we know what is the actual perception of actual cell responsible for perception of light. It is these rods and okay, wait. Yeah, it is this rods and cones, right? But the rods and cones are not present on the surface of this retina, right? They are instead present towards this bottom surface of the retina, right? So this light is actually crossing these layers and it is being perceived by these rods and cones that are present on this other side of the retina. And then that signal is further going in this direction, right? In this direction to the optic nerve, and it is going like this. After that, once it has been received by this layer, it is going upwards to the optic nerve, which is taking it to the brain, right? So this is a characteristic feature. I am not sure if there is a definite understanding of why this is, but what we hypothesize is that the rods and cones, they are very sensitive, and so that there is no overstimulation by photons that are just being scattered. That is why they are deep seated. Also, them being deep seated is protecting them in a lot of cells, in a lot of sense, from any damage that is going to happen on the upper layers of the retina, right? So that is like the hypothesis that we have. But this is an intriguing feature that is present over here in retina, right? So that is why I wanted to share that too. Then let us also read more about this retinal layer. So there is already a stage of the neural computation that takes place at the retina itself. What is this computation that is taking place over here? Let us read about it. So it happens via bipolar cells, right? So as you can see, there are these rods and cones that are present, right? You can see they are connected to these bipolar cells, and in between these bipolar cells are connected among one another by these horizontal cells, right? So what are these bipolar cells doing? They are in the retina are a type of neuron that behave in one of two ways, right? Either they will detect light areas on dark background, right? Or they will detect dark areas on the light background, right? So one of the two things they will do, and if they detect light areas on dark background, they will be switched on. If they detect dark areas on light background, then they will be switched off, right? So this is one kind of processing that they are already doing. After that, what will happen is that these retinal ganglion cells that are present over here, that are basically these bipolar cells are leading to these ganglion cells, right? And these ganglion cells among themselves, how they are communicating via these amacrine cells, right? Now, these ganglion cells have a further more complex set of on and off properties, right? What does this complex set looks like? Most of these retinal ganglion neurons have a particular characteristic response to light that is termed as center surround receptor field, right? So earlier we have seen what is a receptor field, right? It is the area at which the stimulus is attained, right? That is, or a stimulus can be felt or perceived, right? That is a receptive field. Now, over here, we first said bipolar cells, they can be on if there is a light stimulus on a dark background, but if there's a dark stimulus on light background, they are turned off. So it is a simple mechanism or a simple form of reception, right? But over here in these cells, retinal ganglion cells, what happens is it is not the case. The stimulus is not that simple, right? So what is the characteristic response? They have a center surround receptor field. We will soon look at that also, right? So what does receptor field mean? It denotes a region of space that elicits a response from a given neuron, right? That is a receptor field. One of the intriguing features of receptor field of the cells and many others in visual system is that they do not respond to light as much as they respond to differences in light across the receptor field, right? So, that is one thing we need to know about our visual system, right? Only if light is shown into your eyes, how much will the light, will the eye respond to that? It will not respond much to it, right? It will maybe become aware, it will maybe, uh, attain some kind of alertness, but any perception might not be built out of that regarding the object that is showing the light, right? On the contrary, if there are changes in the light stimulus or differences in the light stimulus, right? For instance, in this particular slide, I can see multiple colors, right? So the stimulus of the light is continuously changing. The one that is reaching my eyes, some is looking at this red color, some is looking at yellow, some is looking at the white background, some is looking at the dark, this black colored, uh, you know, words that are written on it, right? So there are some changes in the light that is being sent to my eyes, and those, because of those changes only, I am able to look at this, right? For instance, if I removed these particular figures, I will still be able to read what is written over here, right? But what if I put all of that in white color, right? Why will my eyes not be able to see that? It is still there, right? Why will my eyes not be able to read it? Because the background is also white and the whatever is written is also white. So there is no difference of the light stimulus, right? So it cannot differentiate between what it is getting, right? It will just get a white light, right? So there will be no outcome of perception or no knowledge that will be gained out of that, right? Then this is what that center surround receptor field looks like, right? So what happens over here in the ganglion cells, retinal ganglion cells, what happens is that the light that is falling in the center of the receptive field may excite a neuron, right? So the light is falling in the center over here, over here, and over here, right? So that is why these two regions of the neuron, when the light is on, will stimulate this particular neuron, right? Uh, Whereas when the light is surrounding the area, when light is there in the surrounding area, like in this condition, in this condition, in this condition, right? So at that time, it will be switched off, right? On the contrary, there are other retinal ganglion cells that have opposite profile, right? On center, off surround cells, right? What it will do is that if light is being shown on the center of the neuron, it will be off. But if they are being shown on the periphery of the neuron, they will be on, right? So this is just a phenomena that these ganglion cells have. You do not necessarily need to understand right now what it does. But it is just another form of, neural processing that is happening at this stage. Then the light over the entire receptor field may elicit no net effect because the center and surround inhibit each other, right? So if, for instance, there was continuous stimulation so that the center was also activated and the surrounding was also activated, right? So neither this ganglion cell will be able to perceive it, nor this ganglion cell will be able to perceive it, right? Because there is overactivation and there is no distinction between these signals, right? These center surround cells form the building blocks for more advanced processing by the brain, enabling detection of, among other things, edges and orientations. Right? So that is how we understand that this is a sharp edge, this is a lighter edge, right? Because we are looking at the edge, right? Okay. Then there are certain regions when we are anatomically talking about the eye, there are certain regions that we need to know about, right? One is the blind spot, the other is the focal region or the bright spot, right? So over here, this one is a blind spot, and this one, the black colored one, is the central fovea or the brightest spot in the eye, right? In the retina. What is a blind spot? Now, output of retinal ganglion cells is related to the brain via optic nerves. We had seen that in that previous representation, right? Here, ganglion cells, whatever output they are giving, they are going to form the optic nerve, and that is going to relay the signal to the brain, right? So that is what they are saying over here. Now, there is a point at which the optic nerve leaves the eye, right? So this is the point at which this optic nerve from all these ganglion cells, it will come, it will combine over here, and it will exit the eye and it will go to the brain, right? This point where this optic nerve is leaving the eye is called the blind spot. Why? Because over here you can see there is a area wherein there is no retina over there, right? There is a hollow structure over here, right? So over here, the area where this optic nerve is leaving the eye does not have any rods or cones. Okay. That is why you will not be able to see anything. If the light is falling on this particular area, you will not be able to see whatever is in front of you or whatever is emitting that light or emitting that light, right? So this is a test for the blind spot, right? I will just explain it. Over here, what you can do, like there will be a recorded session also that I will upload. What you can do at that time is, you can, like this is a line that is drawn. On one end, you can see a circle. On the other, you can see a purple colored plus mark, right? If you keep your laptop screens or your phone screen, a bigger screen is actually preferable. If you keep the screen around 50 centimeters away from you and you focus from your left eye towards the plus symbol, right? So you close your right eye, from your left eye, you will focus towards the plus symbol, right? And then slowly you will bring the laptop closer to you, right? So when you are only looking at the plus symbol also, in your peripheral vision, you will still be able to see the this um, circular structure, right? Uh, yeah. So you will still be able to see that circular structure. But as in you are bringing the laptop closer to you, right, from that 50 centimeter distance up till, just next to your eyes, you will find that there will be a region that will come, a duration interval that will come, wherein you will be focusing from your left eye on that plus symbol, and you will not be able to look at that round colored object in the periphery, right? And it will just be some region in which that will happen just before that, and after that also it will be visible, right? That region is your blind spot, because over there what will happen, that the light from this circular object is falling on your blind spot, right? The spot where optic nerve is exiting your eye, and you are only using one eye, right? So the other eye is also not compensating for that phenomena. So that is why that will be a blind spot for you. You can try it later. Yeah. Then, as I mentioned, there is another spot in the eye, which is fovea, right? So this fovea is over here, a little above the optic nerve region, or a little above the blind spot, right? It is where the highest concentration of cones is there, right? The level of detail can be perceived over here is the greatest, right? So if light from certain object is falling at our fovea region, right, that will be the region that will be able to detect or look at a particular object in great detail, right? Because maximum number of cones are present over here. What does maximum number of cones mean? That maximum number of combinations of colors you will be able to perceive, even when if there is a minor difference between orange and light orange in that particular image. Even if it is very minute, the difference, you will be able to spot it, because those many different kinds of cones are actually present over here that will be activated at different wavelengths, that your colored perception will be very, very strong over there, right? So if any of you is from a engineering background, or you are technically sound, you will know that I have recently only been told about this phenomena of the 8-bit versus 10-bit display, right? This is a normal kind of thing that happens in your phone screens, in your tablets, in your laptops, right? So in your 8-bit display, what will happen is the color perception will be fewer, right? So there will be lesser number of bits that are dedicated for the colors, right? So if you open a gradient image, what will happen? You will see some cracks in the gradient, right? That will be visible. But in a 10-bit display, because more number of bits are performing that function. Again, I'm not that technically sound, so I might not say the correct terminology. So because more number of those are at play, what will happen is that uh, the gradient in which you are seeing breaks in the 8-bit display, you will not see that anymore over here. It will be a smooth gradient because those colors that are missing from this 8-bit one, right? Even those colors you will be able to see in the 10-bit display, right? So because again, more number of bits are dedicated over here, there. Similarly, more number of cones are dedicated over here, right? So you will see whatever you are trying to visualize in great detail, right? So the rods are more evenly distributed across the retina, but they are not at all present in the fovea region, right? The fovea region has only cones. Okay. Now we'll talk about the visual pathway of the brain. Just a moment. Let me switch on this. Yes. Um, So now let us look at how this particular information is being relayed from these cells in the eye, right, that are receiving this information in the form of light, converting it into some kind of electrical stimulus that is going to the brain, right? How is it being processed by the brain, or what is the path it is taking to actually reach the primary visual cortex in the brain, right, or other visual areas in the brain? So the dominant visual pathway in the human brain travels to the primary visual cortex at the back or the posterior of the brain via a processing station which is known as LGN or lateral geniculate nucleus. Right. So these are both terms that we have gone through earlier, right? When we were looking at tracts or we were looking at development and all, right? So we have gone through what is posterior, what is LGN, where is the visual processing area, etc. Right? So over here, what this statement means is that there are multiple pathways through which a particular, stimulus can take to reach the brain. But mostly or most dominant pathway actually is this one, in which the stimulus is being carried forward to the back of the brain, right? Which is a primary visual cortex area, and it is going through this LGN, right, in the middle of the brain, right? Yeah. So this is what this particular pathway looks like, right? It is known as geniculostriate pathway, right? And this is what it looks like. So the neural representation in the lateral geniculate nucleus divides up information on the retinal surface in a number of interesting ways, right? Over here, you can see this is the LGN. And you can see over here, whatever information it is receiving from the eyes, it is further dividing it up into some subdivisions, and then that is actually going and reaching the primary visual cortex area, right? So there's some division of information that is happening over here, right? The objects in the right side of the space, that is over here, this peach colored object, uh, which is termed as right visual field, fall on the left side of retina of both the eyes, right? So if, for instance, I have this bottle kept on my right side, right? So both my eyes will be perceiving this bottle from its left end only, right? If it was, however, kept on the left side, both the eyes will be perceiving it on their right end, right? That is what happens. And because they project to the left lateral geniculate nuclei. So because both of them are perceiving it on the left side, right? This peach colored you can see. So both of them are taking it to the left lateral geniculate nucleus, right? Then this information is segregated into 6 different neuronal layers of this structure, 3 for each eye, right? So then there's a division into 6 different neuronal layers, right? Three for this particular eye's information and 3 for this particular eye's information. The layers of lateral geniculate nuclei are not divided according to the eye, but contain a further subdivision, right? This is not the only kind of subdivision that is there. There are some other divisions also. The upper 4 layers have small cell bodies and have been termed as parvocellular or P layers, right? Whereas the lower 2 layers contain a larger cell body and have been termed as magnocellular layers or M layers, right? So there are 4 small cell body layers, 2 large cell body layers, right? So there will be some reason why it is like that, right? That we will study. So the parvocellular cell bodies, cells are respond to detail and are concerned with color vision, right? So that they are the ones that are concerned with color vision, and they are ones that are going to look at everything in detail, right? So that is why more area is also dedicated to them, right? 4 layers are dedicated to this parvocellular cells. The magnocellular cells are more sensitive to movement than colors. They have larger cell bodies. They are not looking at details of things. They are looking at larger things like movement, somebody's passing by, so your eyes can obviously see that also, right? Maybe if it will not be that temporally sound that it will not be able to see always what exactly passed by, but you will know that okay, something passed by, that something was red in color, something like that, right? And they will respond to larger areas of visual field, right? Apart from that, more recently, there has been a third type of cell, which is known as K cell or conio cell, that has been documented in this lateral geniculate nuclei. And it lies between the magnocellular and parvocellular layers. What does it do? These cells show much less functional specificity than magno and parvo cells and have a different pattern of connectivity, right? Then there were these scientists who were working on a federally, funded project in US. Uh, Torsten Wiesel and David Hubble. Torsten Wiesel was a Swedish neuroscientist and David Hubble was a Canadian neurophysiologist. And they used to work with, cats and monkeys, right? And they wanted to understand how different decisions are being made in the brain, how they are perceiving things, how are they visualizing whatever is being given to them, right? All that was being determined by these 2 scientists, right? So, as I said, they were in a federally funded project, right? And in 1981, they were also awarded the Nobel Prize in Medicine, right? Why? Because they, during their course of study on their subjects, that were the cats and the monkeys, where they were trying to study the visual responses on a neuronal level, what they found was they elucidated the properties of neurons in the primary visual cortex area, right? So what they actually did was, let's read about it a little, right? So they had been, before that, let me give a disclaimer. So what they were trying to do was actually, they were trying to show, they used to make these cats or monkeys sit in front of a screen. They used to continually record, they had some electrodes implanted in their brains, and they used to record the responses that were there in the brain, right? And they used to show them on different kinds of pictures and then they used to look at the responses, right? But they were not able to elucidate like which particular area is, responding to which particular kind of stimulus that they are showing. That was not very, appreciably different over there, but they were not able to record it at that time, right? Then what they found was that one day, uh, at that time, there used to be those other kinds of projectors. Like today, we have these digital slides, right? Let me just go back and just talk about their experiment first. So now we have these digital slides, right? We make them on the computer, we run them on the computer, we do next, it skips on to the next slide, right? Earlier, the kinds of projectors used to be different, right? They used to show or throw light. There used to be a lens. They used to throw light out of that lens and it used to be projected on a wall or on a white background, something, right? And what used to happen was, in between this light and lens, we used to keep like paper sheets in which we had drawn our slides, right? If I wanted to present this, I would have to draw this entire thing on a sheet of paper. It used to be that transparent sheet, transparency, if you know. So you will draw it on that and you will put it inside this particular manual projector, right? And if, for instance, you are presenting on a stage, right? So the projector is far away. So you cannot really, manually go to the next slide. So once you are done with a slide, you have to already know what is there in the next slide and you need to say to the person who is, there with that projector, you need to indicate to them that, okay, next. So that person will manually change the slide, right? And then you will go on to the next slide and then you will start discussing about it, right? So that used to happen. So when they were performing these, they were trying to show these stimuli to the cats and to the monkeys. They were also doing it via a projector. They used to have glass slides in those projectors. Right. And what accidentally happened one day was that while they were recording, again, they were not getting much of a difference or much of a, response that made any sense, or it stimulated one particular area, or that they would know that these cats, mice, monkeys, etc., were even looking at these stimuli that they were showing them. Right. Once what happened, by the mistake of the person, one of these scientists who was changing the slide, what happened? They changed it such that it went far ahead. Right. So if you put something a little further, what will happen is you will miss half the slide. It will go outside of the projector, and instead on one side, what you will see some empty space. So there was the edge of that slide, and there was some empty space, right? And when the cat that was viewing that saw that particular edge, right? Its brain, like in its brain, a single neuron started firing again and again, right? And then they found it very interesting. Then they tried to replicate the same experiment with other of their feline subjects and their monkeys, and they saw that this is exactly what happened in all of them, right? So they uh, then studied that particular, neuron that kept firing for, a very long time. And that is the study of that actually led to this Nobel Prize that was awarded to them in 1981, right? Let us read briefly as to what they have done. What they found was that particular neurons in the visual cortices of the cats and monkeys and the areas in their brain responsible for processing visual information didn't respond to simple points of the light, but rather to lines, and in particular lines and contours with specific orientations, right? So they were not responding to objects and all, but edges, contour like pattern, all this is what this visual cortex area was actually processing, or it was able to respond to, right? Then some neurons also responded to horizontal lines, others to vertical lines, and still others to orientations in between them, right? So what is the kind of line that you are showing to them? What is the angle of that line? Based on that, there were different neurons that were firing in their system, right? In addition, they found that neurons responding to signals from both the eyes, but usually one of these would dominate over the other, right? So, as I said, if I'm looking at this water bottle that is placed on my left hand side, right? Both the right side areas of my right as well as left eye, both of them will have that right side area which will be stimulated by looking at this object on the left, and both of them will take that information to the LGN region in the brain, right? But not both the information will be processed. One of this information will dominate over the other information, and that will be processed to the full faculty, right? Then, over subsequent years, Hubble and Wiesel refined their understanding, mapping the visual centers of their feline and primate subjects with increasing precision. Right? So they were able to map these visual centers now by these experiments, by showing various kinds of objects, various kinds of orientations of these objects. They were very precisely, they were able to map out the visual, cortex area. They found that visual cortex consisted of narrow columns of cells organized by eye preference and response to orientation, which they termed as ocular dominance columns and orientation columns, right? They they formed an elegantly organized functional map of neurons that could process the complex input arriving from both of the animal's eyes, right? So, this is what it would look like a little, right? So, V1 is one of the primary visual cortex area, and there are secondary visual cortex areas also that are present, such as V2, V3, V4, right? So, over here, you can see that a simple cell is there in V1, but this simple cell, instead of responding to one simple stimulus, it is collecting stimulus from various other neurons, and then based on those collective signals it will basically or combination of responses from different cells with centers around properties, they will basically form a response. So just a classification of these cells I want to deal with. What are the simple cells? They respond to light in a particular orientation, right? What are complex cells? They are cells that respond to light in a particular orientation, but do not respond to single points of light. They will respond to multiple points of light coming at the same time, right? Then there are hypercomplex cells, right? They will respond to particular orientations and particular lengths of light, right? So there are certain convergent connections which allow cortical neurons to abstract complex information from simple patterns, right? So if you go back to those illusions that I was showing you earlier, right? That there were some random, these shapes that were spread across, and despite that what you were seeing was complete figures, right? So it happens because of this kind of processing, right? So these are secondary visual cortex area, V2, V4, right? Over here you can see that, for instance, this set of neuron is bringing this information, this is also bringing the same information, and this is also bringing the same information, right? And these are kind of converging maybe at the same location, right? Or this one are different, these two at the same location, and different other shapes are also being brought at that particular location, due to which these particular neurons are able to form a shape in their head from the perception that they are getting from these different kinds of neurons. So scattered signal is being obtained, but the brain is trying to combine all the signal and make sense of it, right? That is happening like this, right? So, yeah. So, for those of you who have just joined, you can just, in the recorded session, you can just go back and check what is the kind of illusion of this perception that I am talking about. We have dealt with it with that particular thing in detail, right? Now, we'll talk about sound, right? So, what I had thought was I will actually complete this entire sensory modality unit today. But since we have just reached sound, I think by the end of this lecture, I will just be able to take it up till sound. Um And the other three are smaller also to cover the auditory, gustatory, and olfactory senses. Sound and vision is the largest one. Vision was the largest one. Then comes sound. The other three are smaller, but still it will take at least 1 to 1.5 hours. So that I will probably schedule one extra class, and I will take it in that so that we can finish with this unit also. Today I'll just finish. I'll take up till sound and I'll finish over there. Right? Yeah. So what is sound? Like we discussed what was vision. Now we want to understand what is sound, right? So what is sound? It originates from motion or vibration of an object. For example, the vibration of vocal cords that are there in my, larynx, that or plucking of a violin string or passing of an overhead aircraft, right? So we have already studied previously that these are a kind of vibrations are creating some mechanical disturbances in the air surrounding us, right? So that the receptors that are responsible for sound perception or sound sensation are mechanoreceptors, right? Because there is physical change in the pressure, or there is pressure difference that is happening or taking place in the air that is surrounding us, due to which we can speak, and due to which other people can hear, right? Yeah. So the human auditory system is capable of detecting a huge range of changes in air pressure from around point triple four triple zero two, no, 4 times 02 to more than 100 pascals, right? So that huge a pressure difference we can perceive. However, the role of hearing brain is not merely to detect such changes, right? So even if we are detecting some pressure differences, is that the outcome that we want to, understand, or that our brain wants to understand? No, right? Our brain wants to make sense of those changes in the pressure that are happening, right? How will that happen? Because it will process that information, and it will try to find in its memory that what does this kind of stimulation sound like, right? Or I heard this particular voice, is it familiar to me, right? Or this person said this thing, do I know this particular sound makes a particular word and what that word is, right? So our brain wants to make sense of that information or that pressure changes. Okay. So, as with vision and other perceptual systems, the goal of hearing is not to create a literal depiction of the outside world, but rather to construct an internal model of world that can be interpreted and acted upon. Right? So, what will happen? This model is constructed not only for ongoing sensory information, but also from previous sensory experiences. Right? So, that is what I'm saying. We have learned over time that if I am saying a word, a particular word, sound, right? What does that word mean, right? What does like if you are hearing this word, you know it because you know the English language, right? If you did not know English, will you understand? If I say sound, will you understand what does it mean? It will just sound like noise to you, right? If I say some word, world, right? It is a German word for forest. But will you understand that if you don't know German, right? So that is what it will also, it is not just hearing something, it is also trying from the past sensory experiences, it is trying to figure out what that sound means, right? For example, we recognize familiar tune when presented in a different key, and we can recognize a familiar voice in a wide range of acoustic environments also, right? So this first example is actually very intriguing. Even if, say, there was a very famous song, right? For instance, any favorites, any famous song. Um, All is Well from Three Idiot, right? If that was a famous song that we are talking about, right? Will even if that was originally played on like 5 instruments, the tune of it was played on 5 instruments, even if one instrument out of it is picked up and the song is played on that instrument, because most of us would have heard that song and that too multiple times, will you not be able to figure out, or will that tune not sound familiar to you? It will, right? Even though it is now the sound that is coming out is different, right? The frequency will be different, the pitch will be different, but the tune will be familiar because the changes in the sound waves that are happening are are somewhat similar, right? So that is how the higher level functioning of our brain works. That is where cognition comes in. So again, if there are multiple things that you are hearing, right? If, for instance, I am trying to take a class over here, I'm sitting very close to a place which is, there's a main road outside. There are some bikes and some cars that are passing by. They are playing horns also. But if I don't concentrate on that, I will probably zone that out, right? If it is not bothering me that much, I will probably zone it out. I will be able to focus on this. It is not that my ear is not receiving that sound, right? It is just that it is not processing it further. So my brain is not, processing that information. So that is why it is not being disturbed by it. However, if, for instance, I was just sitting on the road, at the side of the road, on the footpath, right, taking this class, the sound will be too high that this particular, uh, other sound stimulus that I am trying to filter in, that will be overshadowed, and that will also cause disturbance to me and to you, right? So, what our auditory system does is it breaks down sound by frequency and other features, and then it passes it forward. And based on that, based on that, we can perceive whether it is the sound of barking or footstep or music or streams, etc. Right? So in our noisy environment also, our ears often encounter several sounds at once, but it is the job of our brain, not our ears, to figure out how many different sound sources are there, and what do they correspond to. This will depend on both the incoming sensory information and learned knowledge about the sounds, right? So this is another example of the sound or auditory perception, right? So if, for instance, one was listening to a familiar song, right, such as The Waving Flag by Khan, right? And there was a 2 to 5 second gap in that song, right? That generally happens also when the DJ is playing. He will intentionally, pause the song in the middle. Or if you go to a concert, the singer will intentionally pause in the middle. Does that in your mind, you will still complete the entire sentence. Sometimes you will shout out and complete the entire sentence, right? So automatically, your mind is completing that sentence, right? Even if you are not shouting it out, uh, you will know, right? If for instance, internet was lost for a while, if I was listening on an FM, and in the middle for 2 to 5 seconds, I did not really receive that frequency that well, and the song continued after this particular dash, right? Will I still know? So yes, when I get older, I don't want to sing. I'm not that good at singing, right? I will still know what is this fill in the blank, right? I will be stronger. And then maybe by then the frequency came back and the song continued. So my brain will automatically fill that gap, right? So that is another kind of perception that is there. So in vision, we saw a different kind of gap that was being filled, right? That we have, that is a very, very, uh, interesting phenomena. But similar things happen in case of sound perception or auditory perception also, right? So in this case, auditory cortical regions are more active during the gaps relative to unfamiliar songs, right? So if we are listening to a familiar songs, our auditory region will be more active if there is a gap in that song. If we are listening to something that is new to us, we anyway don't know what is going to be the following statement. We have just missed it. So we have missed it. We can't really recall it from our memory because it is something new. So at that time, it will be less active. Our cortical area will be less active, right? Because it is not working towards recollection also. So our musical and lyrical knowledge can fill in silent gaps which are heard in songs, right? One difference that does exist between auditory and visual senses is their sensitivity to temporal and spatial information, right? Now, when we were looking at techniques that were used to study the brain, we have seen what is temporal information, what is spatial information, right? What is temporal? Temporal basically means something that has to do with time, right? So low temporal resolution means that it will take a lot of time for something to happen, right? High temporal resolution means it will be very fast. Similarly, low spatial information means that maybe I will not be able to figure out where something is kept or where some sound is coming from. High spatial information means I will be able to exactly point at where this problem is, right? Where this particular thing is kept, where this particular sound is coming from. That is spatial information, right? Where is spatial and when or how fast, that is temporal, right? So in case of auditory and visual, what do you think? One will have a higher spatial resolution and which will have a higher temporal resolution? So actually, the higher temporal resolution is the auditory stimulus or auditory perception, and the higher spatial information is the visual perception, right? So the auditory stimulus system is exquisitely tuned to detect temporal information, such as rapid changes in frequency that characterize certain speech sounds, and in grouping information together over time, such as extracting melody from the music. Right. So, for instance, if I was speaking something, right? Uh, you will be able to make sense of my whatever I have said maybe earlier than you will be able to lip read what I am saying, right? So that is like one example of this high temporal information. So different time intervals associated with dots and dashes in Morse code also are much easier to process when heard than read, right? So Morse code either you can, symbol it in a sign language, you can, say or spell out this Morse code to somebody by hand gestures or by blinking your eyes, right? Or at the same time, you can do it via, some knocking, right? Or banging some particular place in that particular order. So it has been tested in real, life that hearing these signals makes a person process them sooner than seeing these signals, right? So, in contrast, it is generally much easier to locate an object in space with vision than with hearing, right? So, if, for instance, I was not hearing, still I will be able to locate that, okay, that is that is where the car is coming from, or that is where the bird is flying, right? But if I just try to hear it, will I be able to really make sense about spatial information of the car because it is passing by, right? I might not be able to understand what it is, and it has already passed by. Similar for the songbird that is flying, right? So and that is where the differences lie. What is the nature of the sound, right? So one of the simplest sounds has a sinusoid waveform, which is when the pressure change is plotted against time, and these sounds are termed as pure tones. Now, what is a pure tone? When there is no interference of any other sound, only one kind of sound you are hearing. That is a pure tone, and it is a very, very, very rare phenomena, right? Even right now, if I lock everything up, I will still have this fan that is working, that is spinning on top of my head, and that at least I will still be hearing, right? Along with whatever movie I'm watching, whatever song I'm hearing, right? So hearing a pure tone is a very, very rare phenomena, right? It is when there is no interference of any other waveform. Only a single waveform for a single sound is present. That is a pure tone, right? Now they have two features that can be quantified. One is pitch, right? So they have a characteristic pitch that is related to frequency of the sound wave that is measured in hertz. What is frequency of sound wave? It is the vibrations per second, right? And the human auditory system responds to sound frequencies between 20 hertz to 20,000 hertz, right? Before like below this, below 20 hertz we call it as infrasonic, and above 20,000 hertz we call it as ultrasonic, right? Both of them we cannot hear. The intensity of sound, that is the amplitude when considered as a sine wave, is related to subjective experience of loudness of a voice, right? So whether I think something is loud or it is soft, that will be subjective, right? And it is the physical property that is associated with it is amplitude of this sound or of the pure tone, right? So this is what it looks like, right? Pure tone will be just one kind of waveform that will keep on going. This is the amplitude. If the amplitude or the peak is higher, then it will be more loud. If it is lower, it will be less loud or softer, right? And what is the pitch? It is this, like in one cycle, how many times this is happening, right? That is the pitch. It is a frequency, right? In perception, sorry. In perception, it is crucial to make a distinction between physical properties of a stimulus and their perceived characteristics, right? So we have already talked about sound perception and how that happens, and what are these physical properties and perceived characteristics? Let us look at them, right? So in vision, there is a close relationship between wavelength of a light, which is a physical property, and color, right, which is a psychological property. So based on what is the wavelength of light, it will activate either the red cone or the green cone or the blue cone or a combination of these, so that similar psychological property, that is color, will be perceived by us. But these two things are still not the same, right? It is possible to see color without its associated wavelength, as in after images, which is a condition, right? And it is possible to also process wavelength without perceiving color, as in cerebral chromatopsia, which is also another condition, right? So it is not always that these things will go in hand. It is possible that the neurons that are responsible for us seeing something in red color are firing. So we are seeing something that is not even red as a red color, right? Or the neurons that are supposed to, perceive this red color cone signal is not working. That is why something that is red, we are seeing it as red also, is not being, stated as red in our brain, right? So both the cases are possible. Although in hearing, what happens is that the pitch is related to frequency of sounds and loudness is related to intensity or amplitude of sounds. We have gone through both of these characters, right? So pitch and loudness are regarded as psychological features of sound, whereas frequency and intensity are the physical properties, right? Although the amplitude and frequency might be independent physical properties of sound waves, the subjective properties most closely associated with them, which are pitch and loudness, are not processed by the brain in a completely independent way, right? So there is some linkage that is there between the processing of something as, oh, this is a high-pitched loud noise, right? There will be some kind of overlap between processing, at least. Although the pathway that is taking these two informations might be different, but the one that is processing that this information is mixing the two up, right? Then in everyday life, pure tones are seldom heard, right? It is very, very rare. However, many sounds can be described in terms of combinations of superimposed sinusoids of different frequencies, intensities, as well as phases, right? For example, musical notes typically contain a series of regularly spaced sinusoids, right? So that is again one example. The lowest component is termed as a fundamental frequency and typically determines perceived pitch of a musical note, right? For instance, if this was a musical instrument that was playing, right? In a musical instrument, for instance, if I'm playing a guitar, based upon what is the, string that I'm using, the frequency of the sound that is traveling, it is changing, right? And these different frequencies will have will be a result of different musical notes that I'm playing, right? So from a single guitar, say this was 200 hertz frequency, this was 400 hertz frequency, and this was 600 hertz frequency, right? All these are converging, and this is actually an interference of all this. That is what I'm hearing as music, right? But the lowest hertz frequency over here, right, 200 hertz, will be termed as fundamental frequency, and I will associate this particular song and this particular instrument to this particular 200 hertz frequency, right? The relative intensity levels of different sinusoid components of musical sound are important for discriminating between same notes played on different musical instruments, and this phenomena is known as timbre, right? So it is not pronounced as timbre, it is pronounced as timbre, right? So this is another example that I was saying earlier, that if you pick up your favorite song and say that song was played on like the original music that was given to that song was on a combination of instruments which contained a violin, keyboard, and a guitar, right? And at this point, you only have a guitar, right? And you play that same tune on a guitar, you will still be able to familiarize with that tone, that, oh, this is that tone, but you will be able to differentiate that how is this tone different from the actual song, right? Because the instrument sounds different, something different is happening, you will be able to make that out, and that property is known as timbre, right? Then how is this information actually going to the brain, right? For that, let us look at the ear, right? So there are three major compartments that are present in the ear. One is the outer ear, the middle one is the middle ear, and the inner portion is the inner ear, right? So these three main compartments are present. What happens is that in this outer ear, which contains the pinna, this portion of the ear, this that is outside this cartilage, or the earlobes, and the auditory canal, right? This canal that is going inside from the pinna. This part is the outer ear. The reflection of sound wave within the folds of pinna and within the auditory canal can amplify certain sounds and are important for locating a sound source, right? So that is what is the function of this particular outer ear region. What happens in the middle ear, however, it converts airborne vibrations to liquid-borne vibrations with minimal loss of energy, right? So this middle ear actually has these three bones that are present, malleus, incus, and stapes, which are also known as hammer, anvil, and stirrup, right? Because of how they look, how they are shaped, right? Which basically transfer mechanical pressure that is being put on this side of the eardrum at the end of the airborne auditory canal to a smaller structure which is known as oval window, which will take this to the inner ear, right? Then the inner ear, this part contains chambers that are important both for sensing senses of hearing, which is this region cochlea that is receiving signal from this oval window from the middle ear, right? And balance, which includes these semicircular canals, right? So cochlea converts liquid-borne sound into neural impulses, right? So this will convert this liquid-borne sound into neural impulses. So there is fluid filled in this cochlea, and it will convert this fluid-filled sound into nerve impulses. How will that happen? We'll study. So there is a membrane that is present within cochlea, which is known as basilar membrane. Right? It contains tiny hair-like cells which are linked to receptors, auditory receptors, right? Sound induces mechanical movement of the basilar membrane and hair cells on it. These movements induce flow of ions through stretch-sensitive ion channels that initiate neural activity and the release of neurotransmitters, right? The basilar membrane is not uniform but has different mechanical properties at either end, right? So this end the property will be different, the scent property will be different. I have another image in which this circular structure has been opened up and basilar membrane is visible over there. So we will discuss about this different properties over there, right? The end which is nearest to the oval window, this end is narrower and stiffer and shows a maximal deflection to high frequency sounds, while the end that is near the center of its spiral shape is wider and more elastic and shows a maximal deflection to low frequency sounds, right? As such, different parts of the membrane are sensitive to different frequencies of sound, right? Then you can also note over here that sounds originating from different parts of space do not stimulate different parts of the membrane. The location of sound sources needs to be inferred from other kinds of information, right? So this is the pathway, the auditory pathway that this information is taking from taking from this auditory nerve to this auditory cortex in the brain, right? This is known as the ascending auditory pathway. It is not a passive transmission of information from here, but rather involved in active extraction and synthesis of information in the auditory signal, right? So there are 4 or 5 synapses in auditory pathway, right? One synapse is over here between auditory nerve and ventral cochlear nucleus and between dorsal cochlear nucleus, right? Then it is happening between inferior colliculus and superior of olivary nucleus, then between inferior colliculus and MGN, then between MGN and auditory cortex, right? So there are multiple synapses that are present in this pathway, starting from projections in auditory nerve to the cochlear nuclei in the brainstem, and ending with projections from MGN medial geniculate nucleus to the primary auditory cortex, which is also known as A1 or the core region, right? This primary auditory cortex is located in Heschl's gyrus. Sorry, I have a hard time pronouncing this word. In the temporal lobe, and it is surrounded by adjacent secondary auditory cortical areas called the belt and the parabelt regions. Right. So this is A1 region. Those will be the A2, A3 regions. These secondary regions also receive some input from the MGN, and hence damage to the primary auditory cortex does not produce complete deafness but leads to problems in identifying and locating these sounds, right? So as I said, this is the A1 region, which is a primary auditory cortex region, and then there's A2 region, which is this region of secondary auditory cortex or the belt areas. Over here, there is one that is belt and another that is parabelt, right? So this is the belt area, this is the parabelt area. If we talk about the number of neurons that are present at the cochlear nucleus level over here, right? Cochlear nucleus level, in the cochlea of the inner ear itself, there are around 90,000 neurons that are present, right? In the MGN, there are around 5 lakh neurons that are present, and in the auditory cortex area, you can see there are 100 million neurons that are present, right? In addition, there are also descending top-down pathways that go as far back as cochlea itself and may be important in auditory attention, right? To understand or to pay attention to somebody in particular. If I'm trying to hear something, some very faint noise and trying to locate spatially where it's coming from, I will hear it very silently, like, right? So over here, you can see that basilar membrane that we had been talking about. Although this particular area of the cochlea is wider, and this particular end of the cochlea that is towards the inside of that spiral is narrower, you can see the basal membrane or basilar membrane that is present is actually narrower on this end and it is wider on this end, right? And we were talking about changes in this basilar membrane only that is happening. So this is a cochlea, this is unwound or unrolled cochlea, right, that we can see over here. So the early auditory system can be said to have a tonotopic organization. What is a tonotopic organization? That based on what frequency is being heard, different kinds of bunches of neurons will be activated, right? Just as different parts of the basilar membrane respond to maximally to different sound frequencies, neurons with auditory nerve respond maximally to certain sound frequencies more than others, right? So over here you can see that this is responding to low frequency noises. This is to medium frequency, and this to high frequency, right? Similar is the case over here in the basilar membrane. Different frequency sounds will be attended by different regions in this basilar membrane, right? So a nerve bundle is orderly such that neurons responding to higher frequencies are located on the periphery, right? So in this, what happens is that on both the sides, there are neurons that are responding to higher frequency waves, such as 19,000, 20,000, 17, 18,000, right? And the ones that are responding to the middle frequency or low frequency are in the center, 1 hertz, 2 hertz, right? Sorry, it is starting from 20. So 30 hertz, 40 hertz, right? To some extent, this kind of organization is also carried upwards to early cortical stages, right? You can see over here in this cortex area, on the periphery is the higher frequency sounds, in the middle is the lower frequency sounds, right? In both humans and animals, there is evidence that central region of the primary auditory cortex responds to lower frequency and outer region on both sides to higher frequencies, as is seen over here, right? So these are certain comparisons that we can do between these auditory and visual systems, right? If we talk about the root, auditory system, the root is through MGN and it projects into primary auditory cortex, while visual system, the root is through LGN and it projects to primary visual cortex, right? Over here, there is a tonotopic organization that is orderly mapping between sound frequency and position in the cortex, while in visual system, there is retinotopic organization, which is orderly mapping between position on retina and position on cortex. Then auditory system is more temporally sound than visual system, while visual system is more spatially sound than auditory system, right? Similarly, if we look at the functional specialization of feature processing, then in auditory system, it is less well documented. There are less well documented domains, and visual system has well documented domains for color and movement, right? If you remember from the previous lecture on Friday, you will also recall that the area that is dedicated on this cortex to the visual system is also actually to the primary visual cortex is also more than that dedicated to the primary auditory cortex, right? It is corresponding to this functional specialization. Then in auditory system, there's evidence for separate auditory pathways for what versus where and how, that what is it that we are hearing versus where is this sound coming from. There might be different pathways that are taken for this information to be processed. While in case of visual system, what happens is evidence for separate visual pathways for what versus where and how is definitely again there, right? So after that, there are three other things that we need to discuss. The other touch, the taste, and the olfaction, right? We'll take them up in a short session. I will try to finish up in 1 hour extra session that I'll plan sometime in this week, and I'll update you guys also on the group, right? Because I will really not be able to finish it this soon. Right, so a great deal is still left. I'll not be able to finish it that fast. So yeah, so that will be the end of this particular session. As I said, I will keep one extra session for summing up our discussion about the sensory modalities of the brain, right? There are three sensory modalities that we have to deal with: the touch, the taste, and the olfaction, right? These are going to be shorter as compared to what we have already dealt with, vision and auditory responses, because more is has actually been known about these. Olfaction also actually a lot is known. So we'll see how much we can cover in one more session, and I'll inform you regarding when that session is, right? So I can just stop the recording. If there's a question, you may ask. If not, we'll just end this.